In this project, we used novel statistical techniques to investigate what drives spring phenology in different species in Alberta. Plants use several different environmental cues to determine their timing of spring phenology. These vary for different regions, but three of the key ones for plants in temperate climates like Alberta are spring heating, photoperiod, and precipitation. And a very simplified way to think of how this happens is when plants meet certain thresholds of each of these variables, they will initiate some sort of phenophase, such as first bloom. However, it's not actually so simple that each of these environmental cues contributes equally at all for different species. And for example, species that bloom earlier in the season, we expect from past literature that spring heating is a more important driver of spring growth than these other factors. But we're interested in knowing the degree to which these different factors affect different species of plants, and this is where our novel technique comes in, as you would typically have to set up an experiment to determine how much something like day length affects the timing of flowering versus how much spring heating does. For this project, we used data from the Alberta Plant Watch Network, which is a citizen science project. Their database has observations of different phenology events from all across Alberta from 1987 to 2016 for about different species, and you can kind of see the spread of observations in this map here. So this gives us a lot of information about how the timing of phenology for different species differs from one another, but also how it varies spatially and temporally. However, as you can see here, what this database lacks in and of itself is the environmental cues we are interested in looking at in this study. So what we did is we used a package from R that allows us to pull data from DayMet, so we could supplement this data table with the environmental variables we're interested in looking at. A couple of the ones we calculated were degree days above zero, which is our measure of spring heat accumulation, day length at the time of observation, and precipitation, and some other just average values. And in order to get this data, we pulled this from DayMet, which is a database from NASA that provides graded daily climate data for the entirety of North America. So the next step in this analysis was to construct predictive models using these derived spring heating and day length values from DayMet. As our measure of spring heating, we calculate the accumulated days above 0 Celsius, as this is how plants keep track of spring temperature. Both models follow the form of if in the year we see a temperature that reaches a certain threshold for a given species, we calculate one for each species, then we predict that the phenological event will occur. So the results of these predictive models are shown here plotted against the observed time of phenophase. As you can see, the spring heating model generally outperforms the day length model in terms of variance explained. We then use these values in variance partitioning to examine how much influence each environmental cue individually has on predicting the observed timing of first bloom. For this, we constructed the linear model with the observed day of year as the response variable and the predicted timing of the two models as the explanatory variables. The resulting variance partitions can be seen in this stacked bar plot, and just a note, the order of the species is based on, from left to right, earliest blooming to latest blooming. From this, we can clearly see that spring heating is a better predictor for all species except for Cornus canadensis, where day length is surprisingly a better predictor. In these plots, it looks like earlier species have a greater amount of variance explained by degree days, and we tested this using a correlation test between the R-squared value of each species and the average day of first bloom, and found that the degree day models did in fact explain more variance for early species than for later blooming species, and we didn't find any such relationship for day length. These results that we see here are actually quite interesting to think about in the context of the literature. In the literature, they suppose for early blooming species, they're more related to spring heating, and this is what triggers their different phenology, whereas late blooming species are less influenced by spring heating, and they're more influenced by photoperiod, so they bloom at a consistent time of year each year. However, our results really don't show this. We see that spring heating is a driver for both of these. And we actually see for early blooming species a trend that spring heating is more influential. They respond more to this, and it's a better predictor. But they both for both spring heating is actually the primary driver. This is interesting when we think about climate change. In the literature, they might suppose that only early blooming species are at risk for having their spring bloom times affected by climate change. However, our results say that actually both of these will be affected by climate change. We mentioned earlier on that precipitation and there are other environmental factors, and these are the next steps for this project, is to investigate those with this kind of methodology.